Okay, again, we're just talking about the types. This is how they're going to be classified, whether they're naked, whether they have membrane fusion, whether they have phagocytosis. The video is long and complicated, so we are not doing it. Replication. When animal viruses remain dormant in host cells, viruses are called latent. They may be prolonged for years with no viral activity, but the incorporation of the DNA is actually permanent. So this is where you'll see people present with something that they actually caught years and years before. It's unusual, but it can happen. It's like an episode of House. Here's just a brief comparison. You can look at that on your own. Why do lysogenic and latent viral infections generally last longer than lytic infections? Lysogenic and of course latent, which are waiting for you, usually involve extended periods of time, days to years, during which there's no transcription. And replication only occurs in host if it replicates. Lytic infections typically proceed directly to synthesis, assembly, release, and viral entry. So that circle goes around and then it's sort of done. So the other ones generally last longer because they incorporate more just into your DNA or RNA. And you may have seen how I keep talking about tumors. So this is really directly related to cancer. So genes for cell division can be turned off or turned on. Neoplasia is the uncontrolled cell division of multicellular animal cells, and they form a neoplast cell, which is called a tumor. So benign versus malignant. Malignant tumors are also called cancers. Metastasis occurs when tumors spread. So if it metastasizes, it means it's spreading. Malignant, it's not spreading. You can just remove it and it's good. So you have a primary tumor formation. You have a micro metastasis, and then that can get bigger, and clinically it can go all over your body. That's what those definitions mean. Basically, uncontrolled activation of oncogenes can lead to cancer. Some environmental factors that contribute to oncogenes. So oncology is the study of cancer. UV light, radiation, carcinogens, viruses are all causes of cancer. So essentially you have your normal DNA, you have your carcinogen or something that you're going to put here. Sometimes it can repress this and you don't have any cancer. Sometimes the virus inserts a promoter here that actually promotes the transcription of this oncogene, and then you still have a repressor here, right? So the gene is turned on, but it doesn't matter because you have a repressor. Now here you have a promoter, you have this gene, you don't have a repressor because the virus inserts into the repressor gene, turning the repressor gene off. See how the repressor actually comes back to the beginning? Without this repressor gene, your result is cancer. Do you see how that works? So this is how the cancer can happen. It can insert a promoter here, and the virus can also insert something into the repressor site, which won't let this tumor be suppressed. Viruses cause 20 to 25% of human cancers. Some carry copies of these genes as part of their genome. Some promote already present, some interfere with tumor repression, things like Hodgkin's, cervical cancer. Those are viruses known to cause cancer. Why are DNA viruses more likely to cause neoplasia than are RNA viruses? I mean, I'll tell you, but I'm not going to ask you. DNA viral genomes are more likely to become integrated into the host and insert foreign DNA. Again, not a question I would ask you, but just so you know. Viruses cannot grow in standard microbiological media. They're cultured inside host cells. 3 types of media for culturing viruses: mature organisms, embryonic eggs, and very specific cell cultures. Culturing viruses usually are lysis. A bacteria produces these plaques, and the plaque assay is organized by phage assemblies. They look like this. Here's a bacterial lawn, and on there you get viral plaques, which are actually viruses. Most of the viruses are cultured in mature organisms such as mice or eggs. There are a lot of ethical concerns with this, right? And this happened a lot during COVID because we were rolling right through these kinds of things to try and come up with a vaccine. So more than normal animals and eggs and things were being used to try and figure out what virus this was and come up with a vaccine. So laboratory animals are difficult and expensive to maintain. Chicken eggs are inexpensive. Fertilized chicken eggs are often used, and most vaccines prepared in chicken culture. This is why if you are very allergic to eggs or chicken, you have to be really careful about vaccines. This is sort of where the misinformation about like harmful vaccines, it was people who actually were having a reaction to the protein from the egg, not necessarily from the vaccine. So if you do have an allergy like that, you have to be careful about vaccines and how they are cultured. Here is generally what happens. They do some injections into the embryo, and this is how they culture the embryonated chicken egg. You could also use tissue culture. So this is what a tissue culture would look like. This is an example of a cell culture from blood. HIV replicates only in certain types of human cells, and one early problem with AIDS research was culturing those cells. Why can we do it now? We are able to culture the HIV virus because a cell type that adapted to that condition and was still susceptible to HIV was developed. It is also necessary to discover what conditions to maintain both cultures and viruses. And when this culture was figured out, we have made leaps and bounds in terms of HIV and AIDS research. Again, I would not ask you that question. That's kind of an advanced question, just to let you know how these things work. Are viruses alive? You know, that's a weird question. Some consider them complex pathogenic chemicals. They do invade cells. They're complex. They replicate themselves. They evolve over time. So maybe. Here is some virins on plants. Some characteristics of prions. They're infectious agents. They are disease causing. They can be infected in meat. What else do I want to tell you? That's really about it. Normal sterilization procedures do not deactivate prions. Prions destroyed by autoclaving. They need to be incinerated, burned, autoclaved, or in concentrated sodium hydroxide. The European Union recently approved the use of enzymes to remove them from medical equipment. This is the biggest problem, is you get them in diseases and in the central nervous system, and it's not good. Here's some comments.
In 1982, Stanley Prusner discovered that a sheep disease called scrapie was caused by a proteinaceous infectious agent, an infectious agent composed entirely of protein without any nucleic acid. Prusner named these new agents prions for proteinaceous infectious particles. Prions do not reproduce like other infectious agents by multiplying or growing. Instead, they cause other normal cellular proteins of the same type to misfold into the infectious form. For his work, Prusner was awarded the Nobel Prize in Physiology or Medicine in 1997. Since 1982, several other diseases have been shown to be caused by prions, including bovine spongiform encephalopathy, also known as mad cow disease. Cells contain a gene that codes for the primary sequence of amino acids for the prion protein, PRP. Normally, PRP folds into a functional form with several alpha helices called cellular PRP. The normal function of the prion protein is not well understood, but it's believed to be important in normal synaptic development and function, and it may be involved in stabilizing the structure of the synapses and in establishing memory. The prion protein is also capable of folding into a form with beta-pleated sheets. Prions do not reproduce like bacteria or viruses. Instead, infectious prions convert normal prions into more infectious prions by folding them into beta-pleated sheets. Thus, the number of infectious prions is increased, but by conversion rather than traditional.